In this video, I'm going to be describing how to download data with Pangea. Pangea is a system for scientists, particularly biologists, to store large-scale sequencing data, large information, share it with other scientists, facilitate bioinformatics analyses, things of that nature. One of the key features of this is being able to download data, and that's what I'm going to be describing in this video, how to do it. So, as just a preliminary, I'd like to describe sort of the key model of Pangea, how Pangea stores data, how Pangea organizes data. And this is all based around samples. Pangea organizes all the data around samples. These can be a single swab, a single blood sample, something like that. Is just sort of what you would consider a typical sample in any kind of biological setting. Samples are grouped into sample groups, which these usually represent projects or like an important sub-project of a larger project. Sample groups, the name is, I suppose, fairly self-explanatory. And then samples and sample groups both have what we call analysis results. Analysis results represent actual data like sequencing reads or then results that are analyzed from that data, such as a UMAP single cell plot or something like that, generated from other data. So this is what we have for our sort of data model for Pangea: samples, sample groups, results. Now I'm going to describe how to download some of this data. So how we download data using Pangea, there are two main ways for downloading data from Pangea. The first way is going to be using the web app. This is the web app we're in right now. And then the second way is using the command line interface, the CLI. The difference here is that the web app is, of course, a little bit easier to use, a little bit more intuitive, but the command line interface is going to be much better for any kind of bulk operation that we need to do additional data analysis, other things with. So we're going to dive right in. We're going to start off showing how we go and download data from the web app. So for example here, I'm going to show how you can download data from the Metascope Consortium. As an example, I'm going to use Metascope Seoul. These are going to be samples from the city of Seoul collected as part of the Metascope project. And here we see the sample group page from Metascope Seoul. We see that it has 80 samples, it's got 3 analysis results, it's got some metadata, it's got some other stuff that we're not really going to talk about in this video. So we're going to dive in, we're going to look at how to download some raw read sequence, or actually some cleaned read sequencing data, from a sample here. So we've got this sample here, we saw before that it was in the Metascope Seoul group, that's how we got to it. And we see that it has this analysis result called cap2 clean reads, error corrected and human free read. So we want to download, we're going to click on that, and then we see that this is 2 fields, clean reads 1, clean reads 2. We just click on those fields and presto, see that it starts downloading. Now, because this is a pretty big download, I'm going to go ahead and cancel that. But that would eventually download this entire read file, and we could click through. We could get the second read. We could get all kinds of other results here. Anything we want, really. And we can do the same thing for the sample group itself. We could download some of the analysis results. And here, for example, this result would give us a table of read counts for different taxa across our samples. We could click that, we could download it in much the same way. So that's how we get data from the web app. That's how we download data from the web app. It's a fairly easy to use interface. It's perhaps a little bit less efficient. If you're trying to analyze a large amount of data, you're going to want something a little bit faster rather than downloading all of these by hand. And that's where the command line interface comes in and how you're going to download data using the command line interface. So to explain that, I'm going to pop over to a terminal right here. You can see that I'm in my downloads folder here. You can be in whatever folder you want that data downloaded to. That's going to be the best way to do this. As a preliminary, the best thing to do, well, you have to do it. It's not just the best thing, is going to be to go ahead and install the command line interface, install the software. You can do that using PyPy, the Python package index, and pip install Pangea API. This is already installed in my computer, so we're not going to have much of anything happen here. It's just going to tell me that it's already installed. But of course, if you're running this for the first time, this is going to install something different. And then once that is installed, we're going to run this command right here. And we're going to just see what options are available, and that command is Pangea API, the name of our command, Pangea API. Download, self-explanatory, sample results. That means the analysis results that are attached to the samples. And let's just take a look at what options are available. And once that loads in just a second here, we will see that there are a few different things we can get. We have email, password. We're going to use that to log in. You can also set those with environmental variables so that you don't have to enter those every time. And then the other most relevant arguments here is that we've got the module name, the particular module that we want to analyze. We have this flag download versus URLs only, which I'll explain in a little bit. And then our organization name and our group name, which is going to help us download the samples. So to get into a little bit of what we need to do, to download those clean reads we saw before, we're going to want to enter our command, Pangea API, download sample results. You can enter your username and password, which this is, of course, not my real password, but you should enter your real password. I just don't want to have mine on the video. And then we want, our first real command, we want to enter the name of the module that we want to download. So for us, the name of that module, we looked at it before, and we'll look at it again, is going to be, once we get back to it, is going to be this cap2 clean reads module. You can, of course, pick any module that's available here. And if we don't specify that, it will just download all of the data possibly can from this organization, which we want. Now we need to enter the organization name and the group name. The name of the organization we're downloading from is the Metasub Consortium. And the group name is Metasub Sol. And we can see that. If we go back to our initial page, you'll see that we've got sample group, Metasub Sol, and that belongs to the Metasub Consortium organization. Depending on where you're downloading data, you may be downloading from a different sample result consortium. So if I run the command, and just a little point here, I'm going to take out my username and password because that's not my real password, and that's going to confuse the command if I give it an incorrect password. For the purposes of this video, know that I've got my password programmed in to work without actually specifying it, but you should put in your real username and password. I'm just going to take those out for the rest of this video. So if we go ahead and run this command, what this is going to do is it's going to start downloading reads one at a time. That's not too bad. That can work for some small files, but typically for big data, like downloading all of the reads from a sample, that's going to be a little too slow. So what we want to do is we want to add this extra command, URLs only. And that's going to give us a list of URLs, and actually it gives us a file name as well for where it thinks that should go, which we can then use to download manually. So that's going to give us a list of URLs. I'm just going to take the first 5 items in that list, which you'll see there would be 160 items in this list normally, since we have 80 samples and both the forward and the reverse read for each of these samples. So this command, once it runs, will give us 5 different URLs, which we can then download manually using a tool like wget. So here we are. These URLs are kind of long, gnarly S3 URLs, and that's okay. So as a demonstration of what we're going to do with these URLs, we're just going to use a command like wget. We're going to tell it to output this file into, we're just going to use the end part of the file name it gives us here. So file name, the sample name that we got it from, the module name, and then the field name within that module. It'll give us a pretty good default sample name. And then the URL itself, which looks like it starts just about here. And we'll see that that's going to go ahead and start downloading, just as we want. Of course, this can be batch scripted. You can pipe and download these things pretty effectively into any program. You can also use programs like Xargs and Split to speed this whole download process up, have a couple of downloads in parallel. None of that's going to be terribly important to what we're doing here right now as well. So as a refresher, what we did is we installed the Pangea API, And then we use this command to download URLs for sample results. So the different parts of this command are URLs only. This tells the command to not actually download the files themselves, but just give us URLs to download them ourselves using a different program, like wget, like curl. The particular module name that we wanted, the name of the organization that we're in, which is Medicine Consortium, and the name of the sample group we want to download data from. Once we had our sample, we downloaded that directly. Once we had our URL, I should say, we downloaded that directly using wget, and then we would end up with some actual data. So that is a quick overview of how to download data. If you're looking for the guide for how to do it, we've been following along in the documentation here at this How to Download Data guide, and you'll see basically the same steps I've run through here: a guide to download data using the web application, a guide to download the data using the command line interface, explaining some of these arguments in a little more detail. And for those of you wondering how you can integrate this into a more complicated program, perhaps a bioinformatics pipeline directly, there's also a little note on using the Python API within a program. That's going to be a little bit more detailed, a little bit more advanced. Thank you for listening, and I hope that this is useful.